cycles and division. What is cell cycles? Cell cycles. Most cells in an organism go through a cell cycle of growth, development, and division, called the cell cycle. The cell cycle makes it possible for organisms to grow and develop, to replace cells that are old or damaged, to produce new cells. There are two main pieces in cell cycle: interphase, mitotic phase. What is interphase? Interphase is the period of cell's growth and development. A cell spends most of its life in interphase. There are three stages in interphase: rapid growth and reflection, or copying of the organelle; copying of DNA, the genetic information in a cell; preparation for cell division. What is mitotic phase? A cell reproduces during the mitotic phase. The mitotic phase has two stages. Stage 1, the contents of the nucleus divide. Stage 2, the cytoplasm divides. The mitotic phase creates two new identical cells. The original cell no longer exists. Cell cycle. The time it takes a cell to complete the cell cycle depends on the types of a cell that is dividing. The cell cycle for some eukaryotic cells might only take 8 minutes, but others can take up to a year. Most of the cells in the human body can complete the cell cycle in about 24 hours. The cells of some organisms divide very quickly. For example, the fertilized egg of the zebrafish divide into 256 cells in 2.5 hours. The interphase stage. The G1 stage. This period of rapid growth, G1, is the longest stage of cell cycle. During G1, a cell grows and carries out its normal cell function. Most cells continue the cell cycle. However, some cells stop the cell cycle at the G1 stage. Mature nerve cells in your brain remain in G1 and do not divide again. The S stage. During the S stage, a cell groups and copies DNA. Strands of chromatin are copied, so there are now two identical strands of DNA. This is necessary because each new cell gets a copy of genetic information. A cell's DNA is arranged as a pairs. Each pair is called a duplicated chromosome. Two identical chromosomes called sister chromatids. The sister chromatids are held together by a structure called centromere. The G2 stage. This is another period of growth and the final preparation for mitosis. A cell uses energy to copy DNA during the S stage. During G2, the cell stores energy that will be used during the mitotic phase of the cell cycle. The cell division, mitosis and cytokinesis. Mitosis. During mitosis, the sister chromatids of duplicated chromosomes separate from each other. This gives each daughter cell the same genetic information. The contents of nucleus divide, forming two identical nuclei. For example, a cell that has 10 duplicated chromosomes actually has 20 chromatids. When cell divides, each daughter cell will have 10 different chromatids. Chromatids are now called chromosomes. Cytokinesis. During cytokinesis, the cytoplasm divides and two new daughter cells form. The organelles that were made during interphase are divided between the daughter cells. Mitosis, like interphase, is a process that can divide into different phases. Mitosis phase: prophase, metaphase, anaphase, telophase. Prophase. During the first phase of mitosis, called prophase, the copied chromatin together tightly. The coil form duplicated chromosomes that can be seen with microscope. The nucleolus disappears and the nuclear membrane breakdowns. Structure called spindle fibers form in the cytoplasm. Prophase in animal and plant cells. Metaphase. The spindle fiber pull and push the duplicated chromosomes to the middle of the cell during metaphase. The chromosomes line up along with the middle of cell. This makes sure that each new cell will receive one copy of each chromosome. Metaphase is the shortest phase in mitosis. It is an important phase because it makes the new cells the same. Metaphase in animal and plant cells. Anaphase. In anaphase, the two sister chromatids separate from each other and are pulled in opposite direction. Once they are separated, the chromatids are now two identical single-stranded chromosomes. As the single-stranded chromosomes move to opposite sides of the cell begins to get longer. Anaphase ends when the two sets of identical chromosomes reach opposite ends of the cell. Anaphase in animal and plant cells. Telophase. During telophase, the spindled fibers that help divide chromosomes begin to disappear. The chromosomes begin to uncoil. A nuclear membrane grows around each set of chromosomes at either end of the cell. Two new identical nuclei form. Telophase in animal and plant cells. After the last phase of mitosis, the cytoplasm of cell divides in a process called cytokinesis. The specific steps of cytokinesis depends on the type of cell that is dividing. Cytokinesis, animal cell. In an animal cell, the cell membrane contracts or squeezes together around the middle of the cell. Fibers around the center of the cell pull together. This forms a crease called furrow in the middle of the cell. The furrow gets deeper and deeper until the cell membrane comes together and divides the cell. Cytokinesis, plant cell. Cytokinesis in plant cells is different. A new cell wall forms in the middle of a plant cell. Organelles called vesicles join together to form a membrane-bound disc called a cell plate. The cell plate then grows outward toward the cell wall until two new cells form. Cytokinesis in animal and plant cells. Result of cell division. Small elephants grow to large elephants through mitosis.